friends, welcome to Biomix. Do you know where the proteins are synthesized in the cell? They are being synthesized in the cytoplasm. Now the question arises that if they are synthesized in the cytoplasm, how do different cell organelles have different type of proteins with them? The answer is that after the synthesis of the protein in the cytoplasm, they are being targeted to different cell organelles. So what happens in the cell? In the nucleus, the DNA replication as well as the transcription take place. Here the mRNA is formed and this mRNA is transported into the cytoplasm where the ribosomes are present. Okay, after that this protein are being synthesized. So here you can see the protein are being synthesized. In the protein, either in the C terminal end or in the N terminal end, they are having some targeted sequence with them. So this targeted sequences are the amino acid, which is targeting different type of cell organelles. There are specific target sequence for each cell organelles. Okay, so here the protein formation can be in two ways. Like it can be co-translational transfer as well as post-translation translocation. Okay, it can be in two ways, like co-translation translocation as well as post-translation translocation. So what do we mean by post-translation translocation? That means after the complete synthesis of this protein will be targeted to different cell organelles. The examples of post-translation translocation is protein transferred in the mitochondria as well as chloroplast. Okay, and the nucleus. Okay, the next thing is the co-translation. What do we mean by co-translation? Co-translation, that means the protein synthesis is taking place by targeting some of the cell organelles. Like for example, here we can see here there are free ribosomes which are present, and here the sequence is present. Here a sequence is present which is targeting the ER, endoplasmic reticulum. So here the protein formation has not completed. Okay, so here this ribosome will come and bind in the endoplasmic reticulum. After binding, after this protein is being targeted, it will be synthesizing the protein. So here the translocation has taken place. That means this protein has got its target, that it has to target this region or it has to enter this cell organelles, as well as the protein synthesis is also taking place. So the example of co-translation translocation are endoplasmic reticulum, Golgi complex as well as lysosome. Okay, so in this today's video we will mainly target how the co-translation translocation is taking place and what are the receptors, what are the proteins present, we will discuss in details. So what happens in the cell? There are ribosomes present. So these ribosomes which are present, they can work with by attaching with the endoplasmic reticulum, as well as they can freely attach with the mRNA and it will form the proteins. If it is working, this ribosome subunit which are present, the large and small subunit will encode the mRNA, it will bind with the mRNA and it will help in the formation of protein. Okay, so this is example of post-translation. In this way also this ribosome can bind, or else this ribosome can attach with the endoplasmic reticulum and it will help in the protein synthesis. It is an example of co-translation translocation. Okay, I hope you have understood this. So what happens in the cell? In the cell there are two types of transfer. Here we can see co-translation translocation and this is the post-translation translocation. So for this it requires a receptor. So for during the co-translation translocation it requires a receptor which is known as Sec61 alpha. Here the ribosome which is present, it will come and bind in this receptor in Sec61 alpha and it will help in translocation of this protein, or it will help in this protein to come inside the endoplasmic reticulum. Okay, so this sequence which are present, so the N-terminal region will have a target sequence with it. So this target sequence which are present, they are mostly hydrophobic amino acid and they are of 6 to 12 amino acid. Okay, so for here we will see how the protein are being transferred to the endoplasmic reticulum, and how through the co-translation translocation it is being targeted. So here there are ribosomes. You can see here there is a ribosomes present, and it has started the synthesis of the protein. So the protein which is being synthesized, it is being started from a single ribosome. At the initial stage, it is not attached with the endoplasmic reticulum. It is attached with the free ribosome. Okay, and after the free ribosome is attached with the mRNA, it started the protein formation. When the protein formation has started, it is having a sequence with it, which is the target sequence. The red color one, you can see it is the target sequence. Okay, if the target sequence is present, it requires a receptor who can identify this protein. Okay, sequence of this protein. So the protein which is recognizing this sequence, it is known as SRP, signal recognition particle receptor. Okay, it will help in recognizing the signal of this target. Okay, after that the translation will be stopped. Here the protein synthesis will be blocked, because it is having two reasons where it can bind. In one part it will bind this target sequence, and another part it will block the small ribosomal subunit. You can see here this is a part where it has attached with this signal sequence, and this is the part where it has attached with a small subunit and it has blocked the part. Okay, and this SRP receptor which is present, it is a ribonucleoprotein. It is a ribonucleoprotein. What do you mean by ribonucleoprotein? That means this receptor is made up of ribosome, or we can tell RNA as well as protein. So it is made up of RNA. This RNA which is present, it is known as 70SL RNA. Okay, and the protein part which is formed, it is made up of 6 types of proteins. 6 types of proteins are formed. Okay, so the proteins which are formed are P54, P19, P68, P72, P9 and P14. So here for example, the RNA which is present, we can see this is the RNA, and this P54 and P19 bind in this region. P54 and P19 bind in the N-terminal region, and in the middle binds the P68 and P72, and in the C-terminal region binds the P9 and P14. So what are the work of the specific proteins? The work of the specific proteins are like P54 and P19 bind to the signal sequence. It binds to signal sequence. Okay, so here which is the signal sequence? This is the signal sequence, and here the proteins like P54 and P19 will be present. Okay, now next, the P68 and P72 proteins which are present, they bind to SRP receptor. Okay, this is the receptor where it binds. The next is the P19 in P14. This P19 and P14 which are present, they bind to small subunit of ribosome. Okay, so in this region, the proteins which will be formed present are P9 and P14. Okay, in this way, when this P19 and P14, it binds to the small subunit of the ribosome, it will help in the blocking of the translation. This is the first step where the translation has started, or the protein formation has started. After the protein has formed, it forms a target sequence. Then the target sequence is recognized by a SRP receptor which binds the signal sequence as well as the ribosome, and here the protein synthesis is blocked, or the translocation is blocked until it has reached near the endoplasmic reticulum. Okay, now we will see some of the parts of the ribosome. Like you can see here, this is a small subunit, this is a large subunit, and this is a portion where the proteins will be transported inside the ER lumen. Okay, So here we have understood how this protein has started, how it has started its motion, how it is being recognized by SRP receptor. Here the SRP receptor was formed, and here the signal sequence has been formed. Translocation has been started by this single ribosome. Okay, so here the recognition took place, and here there is a translocation inhibition in this portion. Okay, after that in the endoplasmic reticulum there is a protein translocator. This protein translocator is a receptor for this protein like SRP. This is known as SRP receptor. This SRP receptor is helping in the binding of this SRP, which is bringing this protein with it, protein along with the ribosome with it. Okay, after this protein translocator, until this it is being closed. When this is being attached with this receptor, the channel is being opened here. So here this red color portion has entered in this region, in this transmembrane region it has been entered. After that the protein synthesis is taking place, and whenever it is getting a stop hydrophobic sequence, it is being released inside the AR lumen. After it has entered in the lumen, the signal sequence has been cut by a cleavage factor which will cut this signal sequence in it. Okay, so now after this is being done, during this time this SRP receptor this SRP protein which was present, it will be recycled. Okay, because now it is not having any work. If it is recycled, it can bring another one protein which can target. Okay, so this system is done by another one receptor. Okay, so what is that? It is this is helping in the recycling of this SRP. Okay, so here the cutting of this protein which is present in the ER lumen, it is done by a factor which is known as RRF, which is known as ribosomal releasing factor. Okay, so here the system which is done, so for this process to be done, what happens, this translocation, now it is being closed. So this opening of the, during this time of opening of the translocator, here the GTP hydrolysis is taking place. Okay, here there will be bounding of a GTP, and here the GTP hydrolysis will take place. Due to this, it will help in opening of this translocator. Okay, so here we can see that this closing. So what is the requirement and of this protein translocator to be closed? This is being closed because what happens in the endoplasmic reticulum, there are calcium ions which are present. What happens in the ER? In the ER there are calcium ions present. So for preventing this calcium ion to be transported into the cytoplasm, here high amount of calcium are present. If this channel will be opened, through this channel the calcium can be transported into the cytoplasm. So for preventing the transfer of this calcium into the cytoplasm, there is a blockage, or this channel has been closed. This protein translocator is being closed. Okay, so here this is being done by alpha helix. This is being done by alpha helix. This translocon which is present, or the translocator which is present, it is having three segments like Sec61 alpha, Sec61 beta, and Sec61 gamma. The Sec61 alpha is responsible for closing of this alpha helix. Okay, so there is a closing of this alpha helix. It is working as a gate due to the presence of Sec61 alpha. I hope you have understood these things. So the next thing which we will see is how this is being recycled, how the single ribosome, these are the ribosomes which are present, these are the common ribosomes which are present, which can work in the free form as well as bounded form with the endoplasmic reticulum. If it is working as a free form, it will work as a post-translation translocation. If it is in the bounded form with the endoplasmic reticulum, it will coordinately work with the SRP cycle. Okay, now here we will see how this process is taking place, we will see in details. So here there was a sequence. Here the ribosome is having a protein with it. In the N-terminal sequence, it is having a signal sequence, and this is being recognized by a receptor which is received by a protein, recognized by a protein which is known as SRP. And after this SRP has bounded it is inhibiting the process of translation. Okay, after the translation has been inhibited, it is being recognized by SRP receptor, where it comes and bound. Okay, during this time the translocation has been closed. After this, there is a GTP hydrolysis. During this time, what happens? There is gain of energy, and through this energy, this translocone is also opened, and the signal sequence has been released. And now it continues the protein synthesis. Here, there is no SRP to block this receptor. To block this small ribosomal subunit, and here the translocation is taking place, and here there is recycling of this SRP. Okay, after it has transferred, after the protein synthesis is taking place, in here a signal peptidase is present. Okay, the signal peptidase help in cutting this signal. Okay, and until the stop sequence is present, it is being translocated. Okay, translated, or the protein synthesis is taking place, and after the complete formation of the protein, the folding is taking place in the endoplasmic reticulum. If you have understand this video about the co-translation translocation, in the next video we will discuss about the post-translation translocation. Thank you everyone.